All right, welcome to Chapter 1 of Biochemistry, in which we'll talk about an introduction to the chemistry of life. In this chapter, we'll cover 3 different sections. The first section is the origin of life, as well as the functional groups from organic chemistry. Next, we'll talk about cellular architecture, and finally, we'll discuss thermodynamics. So first, let's start with the origin of life, as well as those functional groups. So to think about how life might have begun, we can think about what raw material of life is built out of. And to get a sense of where to begin with that, we can look at the most abundant elements that are available in the human body, which is on this table here. So you can see that the most common element is carbon. And by dry weight, that's about 61% of the body. And before we go any further, let's think about why we are talking about dry weight. Why not just the complete weight? And as you probably know, the human body is mostly water. So if we were to use the total weight, not just the dry weight, hydrogen and oxygen, the 2 components of water, would be massively overrepresented as the elements available. So here we just want to consider the dry weight. So carbon is 61.7%, and then we have our next group of elements: nitrogen, oxygen, hydrogen, calcium. These 4 are in sort of intermediate category, and then we have a number of others, including phosphorus, potassium, sulfur, etc., that are relatively abundant, but in a little bit lower numbers. So those are the raw materials that were available when life began. So once you have those raw elements, those can start to combine together into small molecules that were available in the pre-Martian atmosphere. So some of those small molecules are water, as we've talked about, as well as nitrogen and carbon dioxide, components of the air. In smaller amounts, there were also available methane and ammonia. So how might these inorganic building blocks that we just discussed have actually come together to form life, which is made of larger organic molecules? So to try to figure out how this happened, Miller and Urey did an experiment in 1953. They combined together some of those inorganic compounds that we just discussed, as well as electricity and UV light. Electricity can be easily available in the form of lightning. UV light, of course, is available from the sun. And they found that when they combined together those things, they were actually able to form amino acids, which are organic compounds and which are one of the basic building blocks of life, which we'll talk about soon. So the Miller-Urey experiment proved that it's possible in that one particular way. However, there are also some alternative theories that have more or less credibility. One of those theories is hydrothermal vents, in which in the ocean floor there are these warm pockets, and that heat, as well as the water available, combined with those small inorganic components, could have also potentially formed lights. There are also some people who believe that life has an extraterrestrial source, which is a theory that's very difficult to perhaps disprove. So now that we know that those simple molecules can be formed just through some very basic factors, including UV light and electricity, how might those simple molecules come together to form complex molecules, which ultimately form organisms such as our human bodies? So first of all, let's think about what biological molecules are made of. A lot of them are polymers. Polymers allow for a lot of structural diversity. So for example, if you think about amino acids, there are about 20 biologically relevant amino acids. And if you have a sequence of 5 of them, that gives you 20 to the 5th power possibilities of different combinations of amino acids that are available. So polymers are a really great way to take a simple, small amount of building blocks and create a lot of different types of structures that can be used to create really complex things like cells and bodies. So for the purposes of this course, we're thinking about biological polymers. And so you might be already familiar with some of the monomers and what polymers they form. So the first monomer that we've already talked about a little bit is amino acids. amino acids come together to form proteins, also known as polypeptides. Nucleotides are another monomer. Nucleotides come together to form nucleic acids, which you might also be familiar with as DNA and RNA. Those are also perhaps called polynucleotides, if you want to talk about just a string of nucleotides. And finally, we have the monosaccharide, or the simple carbohydrate. When these combine together into polymers, they form monosaccharides become polysaccharides, or complex carbohydrates. So how do monomers come together to form polymers? Many biologically relevant monomers do this by using a condensation reaction, which is a reversible reaction. So one biological molecule, in this example we have this carboxylic acid on the left, combines together with this amino group on the right. The hydroxyl and the proton get removed in the form of water, and then the carbon and the nitrogen bond together in this condensation reaction. And so now these two monomers have become this single polymer. The reverse of this reaction is called hydrolysis, in which you add back that hydrogen and that OH group, and then you can break down that polymer into monomers. So once we have polymers, which are macromolecules made of a number of different monomers, like you see here, it's often important that they're able to interact with other polymers that are in the same similar location as them. And so the way that polymers are able to do that is with the interactions of complementary functional groups. And the example given here is an amino group, which here has a positive charge at biological pH, and that interacts through an ionic reaction, an ionic attraction to this carboxylate group, which is negatively charged at biological pH. So these interactions, this complementarity among molecules, can allow some polymers to self-replicate or to act as a template for replication. Excuse me. And you might even know which polymer I'm referring to here, so you can think to yourself for a second, which polymers do I know about that act as a template for its own replication? And the way that these polymers can interact are either through complementary shapes, as well as through those ionic attractions like we talked about in the previous slide. So next, as part of this origin of life discussion, we're going to review organic chemical functional groups. You should have already learned these in organic chemistry, and hopefully they are mostly still familiar to you. Here is a list of some common biological functional groups, and before you go and look them up or open up your textbook, I recommend that you just go through this list and ask yourself, do I remember what this functional group is? Maybe even try to draw it out. And once you've done that, go ahead and progress through the rest of this lecture. So we'll start by reviewing functional groups. I've tried to group them here by similarity a little bit to hopefully help jog your memory. So first we'll talk about amines. Amines have a central nitrogen. That nitrogen can be bound to 1 R group, 2 R groups, or 3 R groups. And that can either have a neutral charge or it can sometimes have a positive charge. Sort of similarly, we have the thiol group. So a thiol has a central sulfur. And so any SH bound to an R group is a thiol. Thiols can come together to form disulfide bonds, which you may be familiar with. So if you have one thiol and another thiol, the two hydrogens can be removed through an oxidation reaction, and then the sulfurs can bond together, forming a disulfide bond. That can also be reversed through a reduction reaction to break down the disulfide into two thiol groups. Next, we'll look at some functional groups that contain oxygens. So an alcohol is sort of analogous to a thiol. As you recall from the previous slide, a thiol is RSH. An alcohol group is ROH, and they behave in somewhat similar ways. And to build off the alcohol, we have the ether group. The ether group also has a central oxygen, but it's surrounded by two R groups. Next, we have the aldehyde and the ketone. Both of these contain a carbonyl group, the C double bonded to O. The aldehyde is located at the end of a molecule. It has one R group, and the other side is a hydrogen. That's the end of the molecule. A ketone is a carbonyl group that's located somewhere in the middle of a molecule. So the carbon is bonded to two R groups. Building off of that, we have the carboxylic acid. The carboxylic acid still has that carbonyl group, and additionally to that, it has an alcohol attached, and that can either be at a neutral or it can have a negative charge, as we see here, and that's called a carboxylate. And similar to a carboxylic acid, we have an ester, where instead of this hydrogen, we now have another R group, where the molecule can continue on. Analogous to the ester is the thioester, where the oxygen is replaced with a sulfur, which builds off that same nomenclature from the thiol, where thi indicates you have a sulfur present. So the next group of functional groups, we have first the amides. So these also have that carbonyl at the center, but not bonded to the carbonyl. You have one R group and one amine. So the am from the amine and the amide refers to the carbonyl plus the amine, and that amine can either have no R group, one R group, or two R groups. A little bit similar to that, another nitrogen-containing molecule is the imine. So imines are a little bit like a carbonyl because they have that double bond. So the nitrogen is double bonded to the R group, and then that nitrogen can be bonded to 0, 1, or 1 R groups on the other side. And like the amines we discussed before, that nitrogen can be neutral or have a positive charge. The next set of functional groups all have phosphorus in them. They all have very similar names, but it's important to be able to differentiate between these three types of functional groups because they all behave a little bit differently in your body. And as you progress through biochemistry and maybe other biology courses, you'll see that phosphate groups have a lot of importance in the biological function. They're structural. They're part of, for example, nucleic acids. They also have a lot of functional purpose for signaling and other purposes. So the first phosphate-containing functional group we'll talk about is the phosphate ester. Which, if you look at it without its R group, is called a phosphoryl group. So the phosphate ester looks a little bit like the ester functional group that we discussed a couple of slides ago, except instead of a carbonyl, a C double bonded to O, we have that phosphorus as the central molecule. Phosphorus is double bonded to oxygen, and then you have that linking oxygen that helps make it an ester that sticks it onto that R group. A diphosphate ester is similar to the phosphate ester, except there are two of them. So you have your first phosphate ester, and your second phosphate ester, and that's bound to that R group. Without the R group, it's called a phosphoanhydride group. And finally, the last one is a little bit different. It's the phosphate diester. Has a similar name to the one we just talked about, but it has a different structure. So the phosphate diester, like the name might imply, is two esters, right? So you have two R groups on either side of that central phosphate group. And that's referred to as a phosphodiester linkage. And it helps stick to two R groups. So it might help to perhaps stick to other molecules together. So at this point now you're ready to answer the Chapter 1 Origin of Life practice questions.